Okay, go, take it away. Sure. Good morning, everyone. You can all hear me okay? Yeah. So firstly, thanks everyone for coming. It's a rainy morning, but we all made it. I don't know who typically speaks for these events. I'm guessing I'm a little bit different. So I'm a scientist. I'm not I grow plants for fun on the side, but otherwise I work in a high-end molecular biology lab. So this is what I'm going to be talking about today, is molecular biology, genetics, proteins. I understand for many of you, you might struggle to follow along, and that's okay. If you have any questions, just interrupt me and feel free to ask, and I'll try my best to address your question, just to make sure I don't lose anyone. So my name's Carl. I'm a staff scientist, and let's see if I got this working. Yeah, so my slides will be on a black background. Does this help? That looks better, right? So I'm a staff scientist at the Salk Institute. Who's been to the Salk? Are you guys familiar with the Salk? So most of you are familiar with it, and probably a lot of you have been there, right? So we're up here on the cliffs, up in the kind of La Jolla area, overlooking the water. We're near the glider port, so sometimes you get the paragliders out. And these are the science buildings on the left and right there, and our greenhouses are kind of at the back, overlooking the Pacific Ocean. So my lab, right? I'm a little bit of an outlier in my lab. My lab does not traditionally study carnivorous plants. But within my lab, so I work under the Professor Joanne Corey, and our lab is interested in this, and this is how plants interact with their environment. So a plant seed, right, depending on where that seed lands, it's going to germinate, and then its growth is completely dependent on the local environment in which that seed found itself. So within this environment, right, you're going to have access to light. Are you shaded by other plants? Are you out in open sunlight? So you'll have competition from other plants. You have water availability. You have nutrients in the soil. You have the presence of herbivores, herbivorous insects or larger herbivores. And the plant needs to sense all these things in its environment and then respond appropriately. And usually for a plant, unlike us, right, if we think something is wrong, we get up and we run away. A plant can't do that. So their response generally is not a behavioral one, rather it's a developmental one. For example, a lot of you will know, one thing which our lab studies a lot is what we call shade avoidance. And you'll see this for the plants you grow at home. If you put a plant in the proximity of another plant, it tends to grow very tall and spindly, right, to try and outcompete neighboring plants. So this is a developmental response to the environment. And then in our lab, we're interested in the molecular mechanisms as to how plants do that. So one of these environmental signals which I'm interested in is mechanoperception. So this is basically sensing touch, right? Physical force. And for plants, you know, these mechanical forces come in a variety of forms. You can imagine, for example, here on the left, a root growing down into the soil and coming into contact with its substrate, and this root needs to navigate through the soil. Salt actually is another example of mechanoperception. So if you remember back to your high school biology, right, when you change salt concentrations through this process which we call osmosis, water will start moving around, right, from an area of low salt to high salt to balance out those concentrations. So in the example of a plant, you can start to have water flowing in and out of cells, causing cells to change their shape. And this is another form of mechanical force. And then on the end here, I have this phenomenon known as thigmomorphogenesis, and this is essentially touch, right? So you can imagine a tree growing out in an open meadow, the wind constantly blowing on it, and it can sense that physical force. And over time, you'll see the trunk thickening and a change in shape of the tree. Right. So these are all examples of mechanoperception. Yep.
the last one there, you know, look at like porcupines, right? How they kind of like.
Yeah. I mean, go out to what is it? Scripps, Ellen Browning Scripps Park, I think, yeah, in La Jolla, and you see these windswept trees there, and you can see how they, when they're up on the cliffs, how they grow to respond to that constant touch. So yeah, that's a good example. So what I'm interested in then, right, is again getting down to the molecular mechanisms of how plants are perceiving this mechanical force. And for this, again, I'm a bit of an oddball in my lab. I turn to carnivorous plants, and obviously not a surprise to this audience, but carnivorous plants, right? They have these very remarkable, I would say, responses to touch, and particularly within the Drosoraceae family, which is the main group of carnivorous plants which I study. So this includes your sundews and, of course, the Venus flytrap, as well as some other species. So here is a picture of a sundew, which I took. So this is Drosera capensis. I'm sure many of you grow it, right? And here, this again, very remarkable plant. These hairs which are coming off the leaf, many people in the field call them tentacles. Sometimes they're referred to as trichomes. These tentacles secrete this little drop of glue, which we call mucilage. The insect is attracted to the leaf, either by volatile secretions or color. It's not really clear. Lands on the leaf or crawls onto the leaf, and then these tentacles, right, it then gets stuck to this glue and starts thrashing around, and these tentacles can sense that physical, that mechanical touch. And then they respond. You can see here in this picture how they're starting to bend over. So they try to bring the animal prey to the center of the leaf. Here's a video. I'm sure many of you have seen this before, but this is a video which I took. So this now is Drosera spatulata. You start to see some of these tentacles move, and then over the course of a few hours, for many Drosera species, you'll see the whole leaf curls over. The insect gets drowned in that goop, that mucilage, and then the plant starts secreting digestive enzymes to break down the soft parts and absorb the nutrients to get those nutrients which it wouldn't otherwise get from the nutrient poor soils in which it grows. So, right, we see this clear response, and in this case, a lot of those signals, there's chemical signals which the plant is receiving from the animal prey. There's also touch signals which it's receiving from the animal prey. So we use this here as a model system. What I'm going to show you now, so there you could see it very dramatically, right, in terms of, now we're almost looking at behaviors of plants, right? In the lab, we can now start to look at the molecular level as well and see that these plants are responding to touch at the molecular level. So what you're looking at here, this now is going to be a video of a transgenic plant. So we make GMO sundews and carnivorous plants in the lab. This plant is expressing a sensor, and this sensor is basically, it's a fluorescence sensor, and it's going to light up when calcium is moving around and is present at high abundance. And so what we can see now, so we're going to place, here's the leaf of the sundew here. A fly is going to be placed onto the leaf. Right, and so here's the fly moving around, and you can see these little bits of green fluorescence in these tentacles lighting up around the fly as it starts touching these tentacles. And then what's going to happen over time, you can see that this fluorescence is gradually subsiding as the plant adjusts to the presence of the insect. So we can see this here. So we can see that this responses to physical touch. So this now is a close-up view of some of those tentacles on the leaf. And we're going to come in and we're going to touch these tentacles with a glass rod. Right, and you're going to see this very rapid, right? You can see that burst of fluorescence, which shows that at the molecular level, right, this chemical signal, in this case calcium. So calcium is a very common molecular signal in plants, also in animals, right? And it will go on and signal for things to happen within cells and within the plant and within tissues. You can see this burst of movement of calcium within the cells. Yeah.
Is that looping right now, or is that?
This is looping. Yes. So this is a loop here. Yeah. And here in this example, so now you're looking at the rosette, and you can see that if you do a light touch to the leaf, right, you'll see that you get this local change in the fluorescence there. We can also start to do things like a harsh touch, and then you start to see these systemic waves of changes in calcium going throughout the plant. Yeah, I think this one is quite remarkable, this video. And this is seen in other plants, I should say. So we commonly work with a small mustard weed model called Arabidopsis. Some of you in the audience may have heard of it. And it's also a small rosette plant, and you will see similar systemic waves of calcium. In that case, if you have and this is work of others, if you have an insect chewing on the leaf, for example, you'll see these calcium waves go throughout the plant. So they're sensing physical damage.
What's the considered a light touch and a harsh touch?
Yeah.
What's considered a light touch?
So a light touch is a gentle tickle of the tentacles, or a tap on the leaf, I think was what you saw before. In the case of what an insect is actually doing, let me just pause this. An insect, for example, I've only ever seen once when I put an insect on the leaf, I've only ever once seen these systemic waves, usually, right? And that would make sense. And by systemic, I mean that this wave is propagating from one leaf to another, if you guys are following me on that. I've only ever seen that occur once with an insect where it was thrashing around a lot, and then you saw the systemic wave through the plant. I'm guessing in the case of carnivory, it's more those local changes which are important, because the bending of the leaf, right? Only one leaf bends around an insect. It's not like the neighboring leaves bend. So these systemic waves here in response to a harsh touch, this is probably more similar to what we see in Arabidopsis, where this might be more of a residual defense response, right? So it senses mechanical damage. Yeah, exactly, right. And then you can, and I should say, and actually I'm going to do some advertising here. I'm not going to go into the details of this today, but there is a YouTube video which I uploaded. So talking about defense, right? These calcium waves traditionally in other plants, they feed into defense pathways. And there's a large body of work out there. And this is work of other people, which suggests that carnivory, at least within the Drosoraceae family, evolved from defense pathways. So we see that at the molecular level. There's certain defense hormones which occur when a plant is trying to defend itself against a herbivore. And basically, these plants have taken those defense pathways, which are recognizing herbivorous insects, and now putting them towards attack to actually catch prey. So if you're interested, Salk Education Outreach and I put together a little video which was released for World Carnivorous Plant Day on YouTube, and you can go and look at that further. Yeah.
Is the systemic one where on the hard touch, is it similar to like an action potential along a neurons?
So how I'm going to show, and I'm hoping to do a live recording of an action potential today. We'll see how the calcium signals correlate with what we consider more classical electrical signals is a little bit unclear in carnivorous plants. If it's a direct correlation, or there's some arguments that the action potentials which we measure is something different. So that's still a little bit of a question mark. But there's certainly correlations there. Yeah, question at the back. So there's lots of other ions. So we'll talk about this. So basically, what carries right here, we're talking about calcium, and this is calcium which is moving internally within a cell. When we talk about electrical signals and action potentials, is the room familiar with when we're talking about action potentials, what that is? Yeah. So this is the classical electrical signal and spike which you see propagating through a Venus flytrap leaf, for example. And as I said, I'm hoping to actually measure one today. So this is a change in what we call membrane potential, right? So you've got a cell, it's surrounded by a membrane, and there's a change in the charge across that membrane, and that's what we'll measure. These changes in calcium, I don't know where this calcium is coming from in the cell. It's probably some internal store of calcium, and then it's getting released from that internal store, and then it goes into the main compartment of the cell, which we call the cytoplasm, and that's what's being measured here. So again, there are different techniques. How they correlate with one another is not clear.
Usually calcium is kept in the plasma reticulum,
before being released through a signal like this?
Yeah, it could be there, it could be mitochondria, it could be vacuole, it could be from the apoplast. Yeah, many places. So it's possible. We just don't know. We don't know. Somehow, though, the plant, right? I mean, you saw these signals, you saw these systemic waves, you saw these very local signals. Somehow the plant needs to work out what those signals mean, right? Calcium is commonly used in response to the environment and different environmental signals. Somehow the plant needs to integrate all that information and mount the appropriate response. And this is a big question mark in plant research as to how plants do that. But again, it's beautiful to watch, and to see this in real time is quite amazing, and to see these rapid responses. So my question, right, is what's the molecular basis? Then you can see that these plants, they respond kind of at this behavioral developmental level, where you saw those tentacles moving, you saw the leaf curling over the prey, and then you saw at the molecular level that you have these changes in calcium signals. What's the molecular basis for that? What is the actual protein, right, or cellular structures which are sensing that mechanical force? So this was the question which we sent out to address, and I was very fortunate to have a collaboration with Ardem Pardapudian's lab at Scripps Research here in San Diego to push this research along. So this was a collaboration with their group. And for this, we turned to what I consider the kind of the big daddy of carnivorous plants. It's certainly my favorite. It's would have to be the most famous of the carnivorous plants. And again, I'm going to play the video where we're all obviously aware of what goes on, but your animal is attracted to the leaf. Again, a little unclear how that occurs, but there's certainly volatiles being released, probably color. It crawls over the leaf, feeding on these sugar-rich secretions, and then it comes into contact with these touch-sensitive structures, which then send, as we've already discussed, this electrical signal through that bilobed leaf blade, that trap part of the leaf, and then that induces closure of the leaf. So this now, right, this is no longer a slow developmental response to touch, but it's a very fast bistable movement, right? So this gives us a unique system, I would argue, to understand how plants sense mechanical signals from animals, right? Because we have this very dramatic, very fast response to the environment. So within the Venus flytrap, right, we have these, you've got your bilobed leaf, and then you have these small little needle-like structures on the leaf. So these are our trigger hairs. And there's a close-up of one of these shown here. And it's the bending of this trigger hair then which triggers this action potential, this electrical signal to go through the leaf. And these action potentials, they typically have this form. So again, electricity in biology, it's caused by charged particles, ions, so charged salts. And you can have different concentrations of salts on one side of a cell membrane versus another. And as these salts move around, so they'll move through things which we call channels, there's pumps. So there's different ways for these salts to cross the membrane. And this moves charge around, right? And then you can see this change in charge, and this is the electrical signal. And typically, when we have an electrical signal through our cells, you get something called an action potential. Same here in this plant, right? So the cell is at some resting, right, charge across its membrane. You then have this, when this electrical signal goes through the leaf, you have this part of the action potential, which we refer to as depolarization. And then various other proteins kick in to get the cell membrane back to its resting state. So we get repolarization and recovery. And then what I thought could be kind of fun for today's audience, I'm actually going to try and do a live demonstration and recording of an action potential. Has anyone seen an action potential in real life from these plants? Someone has, right? You're okay. So we'll try it. This is, as I said, it might fail. Usually it works. So we're going to measure a Venus flytrap action potential. I'm going to use this. I put up the URL here. There's this little kit which they sell mostly for high school students from this company called Backyard Brains, and it's kind of neat. So it's a simple kit. So I'm going to stop this screen share, and I'm going to share now with the spike recorder. All right, so this is what their software looks like. For this part of the talk, can we just turn up the lights again? Is that okay? Just so I can see what I'm doing here. All right. On, thanks, just for this part of the talk. Okay, so what we're going to do, there's basically, there's this little device here, and we have one electrode, all right, this electrode here in black. I'm just going to stick that in the soil, so that's going to be our reference electrode. And then here I have another electrode. It's basically just a little piece of silver wire. And this I'm just going to put on the back of the Venus flytrap leaf. And so it's basically, it's going to measure the change in the electrical field around the leaf as current moves through the leaf. To help it detect what's going on, I'm just putting on a little bit of a conducting gel. So we'll just put that on here. At the moment, I should say what you're seeing on the screen here, this is just my speaker. So it's not yet on. So I'm going to bring that other electrode, right, into contact with the back of the leaf. We'll turn it on. All right, so here, right, you're looking at now the electrical field of the leaf. So we've got some kind of resting, right, charge of the leaf. And then we'll see what happens when I touch a trigger hair. So I'll pause that and we can replay it. So that's in real time, right? So let's play it again. Playing here. Seem to have lost it. Oh, there we go. Right, so here was our action potential. Right, so you can see very nicely, and this looks almost identical to an action potential which would travel through our own nerves. It's a lot slower though, right? There's different ion channels involved. It's very, very large, which is why it's so easy to measure, and you can get these kits which bring it up looking beautifully. So there's some differences, but the shape, right, is basically the same, and the principles. Yeah, right. It's again, it looks like what occurs in our own nervous system.
So was that just one trigger?
That was touching one trigger hair. All right, so we can do another one. I'm just giving a little bit of time. Obviously, in this room, we're aware of the two-touch, generally the two-touch rule. If I touch it again in short succession, what's going to happen is that trap's going to close and it will break the connection with the electrode. But it's probably, all right, we can probably do it again now. So let's try once more. This is real time. Yeah, here we go. Very hard to see. Yeah. So I did a double touch that time. It just closed. So that's why the signal is has now, it's detached from the electrode. But you saw again, you had that change in the electrical field going through the leaf. And in this case, as I said, I did a double tap there. You saw kind of the double spike and the leaf just closed up on me. So that's what it looks like. I'm going to switch back to the slides now.
So the Pensis or Procko, does it look the same or similar to that when they're?
Yes, so they also have So the question was, in Drosera, does it look similar, right? And the answer is yes. I've never measured from, I don't know who asked the question, so I'll just at the back. Yeah, so I've never measured from Drosera. Obviously the tentacles there and the leaf, they're a lot smaller, so it's just, you need a little bit more sophisticated equipment to get in. Generally that action potential travels right from the head down the stalk and then stops before getting to the base. It stops then at the base and doesn't generally propagate much further than that.
And do these change depending on the cultivar, like if it's a B52 or if it's a smaller typical?
You know, I don't know. I don't know the answer to that. Yeah, whether or not in a larger plant, if it takes longer to propagate, I really don't know. Yeah, maybe. So again, right? So that's what the action potential looks like. We have this resting potential. You get this spike as that electrical current passes from one cell to another, and then those membranes return back to baseline. And basically, that electrical signal then is being propagated from cell to cell to cell. And travels through the leaf and eventually meets the midrib, and then it's not really clear what's going on at the midrib, but then that you get two of them, right, in a brief period of time. This causes a change actually in calcium levels, and then those calcium levels, they keep climbing up. It passes a threshold and you get closure of the leaf. So these electrical signals, right, again, in plants, in animals, right, any example from biology, electricity involves charged salts, these ions, and moving these salts right from inside the cell to outside the cell, and therefore changing charge across the membrane. So what we're interested in then, right, we're trying to find something which can sense mechanical force and therefore alter salts at the membrane. And to try and find then the molecular mechanism of what's going on in these plants, what we did is a very large gene expression experiment. So here we took these trigger hairs, basically we cut the trigger hairs off thousands of leaves, and then we can extract, in this case, what we call the RNA, right? So this is a product from genes. We extract that from these plants, and then we do a method which we call RNA sequencing, and basically it determines, right, what you're looking at here on the left, is essentially to see which genes, right, within the genome are turned on and off, right, and how highly they're turned on. And so here, basically anything on this y-axis, which is, right, these dots which are up high here on that y-axis, these are the genes which are more differentially expressed. Basically, they're turned on more within the trigger hairs. And so these are the genes then which we're interested in, because potentially then, right, these are involved with certain processes which go on within the trigger hair. And three of them stood out to us in particular, and mostly this one here, which we called FlyC1 or flycatcher 1. And you can see here on the right, this is showing, again, the expression of that gene, FlyC1, also a related gene called FlyC2, and then this other gene here on the right, which we call Oscar, looking at its expression in the trigger hair relative to the leaf. And the reason these genes stood out to us is that these code for ion channels, right? So these are things which can be gated, open and closed, and when they're open, they can allow the free flow of ions from one side of the cell membrane to the other. So you can start to change charge. So we're interested in these three particular genes, and in particular that flycatcher 1 gene. Here what you're looking at now, this is a scanning electron micrograph. You might have seen some of these images before in books. This now is looking at the base of the trigger hair. All these kind of balls on the side, these are the digestive glands, and then each of, right, these are cells of the digestive glands coming here to make kind of those little balls on the leaf. This is the trigger hair coming up here, right? So that's the lever, and then down we have the podium, and then between the podium and the lever, you have kind of these crenellated cells there, and we call this the indentation zone. And this is where, when an insect bumps into the trigger hair and bends it, this is where maximum flexure of the trigger hair occurs. So this is the sensory zone. This is where the sensory cells are located. And what we find, so here you're looking at results from this flycatcher 1 gene, FlyC1, is that you can see that this particular gene is expressed specifically within those sensory cells. So this is a different type of experiment, and this was conducted within Ardem's lab by a postdoc there, Bill Keenan. This now is what we call a cross-section through the trigger hair. These are the sensory cells here. Let me, for the Zoom recording, let me circle them here, right? So this is the indentation zone. These are the sensory cells, kind of these elongated cells, which will be bent here. And then what you want to look at here in this particular technique, all these little red dots in here, these correspond to that, the product of that particular gene, the flycatcher 1 gene. And these little red dots, they essentially appear exclusively within that sensory zone. Right, so this gene is expressed within the right place to be involved with touch sensation. And then what we also find, right, so now we're doing a comparative experiment with Drosera. Someone was asking before about action potentials in Drosera. And I was saying how that action potential, right, generally starts around the head or neck region and then travels down the stalk. It's not totally clear what the sensory cells are within the Drosera tentacles, but it has been hypothesized in the past that it might be these outer cells on the head of the tentacle. So again, here you're looking at a cross-section of the tentacle. We have these outer cells here. These are the secretory cells, but it's also been hypothesized that they might double as the mechanosensory cells as well, sensing the touch of the insect. And we also see here in this experiment, right, that the equivalent genes in sundew, that these are also expressed within what is very likely the mechanosensory cells of this plant as well, right? So this gene and its related gene, flycatcher 2, they're expressed in the right place to be involved with touch sensation. And the reason we were particularly interested in these genes, the flycatcher 1 and flycatcher 2 gene, as well as the Oscar gene. So FlyC1 and FlyC2, what we knew about these genes and why we're excited is that they are very similar to another protein called MASGS. And so MASGS was identified in bacteria. It's a bacterial gene, and it codes for a mechanosensitive ion channel in bacteria. And what happens in bacteria, right? Here's the MASGS channel depicted here in the cell membrane. And then if you get stretching of that bacterial cell membrane, right, it will open that ion channel. It will gate it open, and it will allow the free movement of ions from one side of the cell membrane to the other. So in bacteria, the role of this channel is essentially thought to protect the cells from what we call osmotic downshock. So if you were to take a bacterial cell and you were to put it in a low salt environment, again through this process of osmosis then, right, water is going to start coming into the cell is going to start to swell. To prevent that cell then from exploding, these channels sense that membrane stretch, they open, they allow the movement of these salts, right, to equilibrate the salt concentration in and out of the cell to prevent the cell from rupturing. So our results then would suggest that this ion channel then has been right co-opted through evolution. Its ancestral role is in osmosensing, and now through evolution, it's been co-opted by these carnivorous plants and put towards this new purpose of sensing touch. And so this is work from, I was not involved with this study, this is from Ardem Patapudian's lab at Scripps, and this is just showing the actual structure of this particular channel, right? So it's sitting here in the membrane, right? That was the pore you first looked at. You can see it spinning here. It has kind of these helices, which are embedded within the membrane and lying parallel to the membrane, and that might be part of how it senses the stretch of the membrane, which then gates it open to allow the movement of salts right through the pore. I got a question over here. Yeah.
Familiar with Adam's work. piezo.
Yeah, the piezo channels. Yep.
So there's a relationship between piezo channel and just fractal.
Yeah, so the question from over here, for those who didn't hear it, is they were just saying that Adem, he's most famous for studying these channels called the piezos, so piezo 1 and 2, for example, in animals. So these are the major mechanosensors in animals. They do have equivalents in plants, but it's a different channel. It's very different from this one. Yes, so yeah, that's right. Yeah, and that was for the discovery of the piezo channels. Yeah.
About the phylogeny of these proteins here, are they more closely related to bacterial enzymes than plant enzymes? Or
They're different from the bacterial channels. They have, for the people in the audience who are in the know, we talk about things like transmembrane domains. They have additional transmembrane domains. So the plant, and they're not just found in Venus flytraps, they're found in other plants, these mass gas light channels. The part which is conserved with bacteria is essentially that pore region, the pore domain, so the bit which crosses the membrane, but then there are differences beyond that.
it's not horizontal gene transfer or anything like that.
It's ancient, right? Yeah. So and in plants, right? This is a family in plants. It's not entirely clear what these channels are doing. It's thought that they probably are having roles in what we call homeostasis, sensing salt as well. The best example of what these channels are doing in plants, I think, comes from another one of my collaborators, Elizabeth Haswell's group, and looking at pollen hydration, right? So when pollen lands on the flower, water starts to come into the pollen cell and it starts to expand, right? So similar story to what's happening in bacteria, and these channels kick in to prevent that pollen cell from rupturing. So what this would suggest then, right, based on all this homology, is that these channels which we're looking at, right, this fly, and in particular the flycatcher 1 channel, which has been our major focus, is that these channels are mechanosensitive, right, and that they're sensing touch, and they're gated by open and closed by that stretching of the membrane. And so this was an experiment done by Swetha Murthy in Adem's group. And basically here she's taking mammalian cells and then she's putting this Venus flytrap channel into mammalian cells. And then she can measure, right, the current which passes through this channel when you start to stretch the membrane. And that's the experiment showing here. So this is the stimulus on the membrane. All right, she's applying some pressure to stretch the membrane. And then you see when you measure the electrical current across that membrane, is that you have kind of this step-like change in current. So these are individual flycatcher 1 channels opening, right, as that stretch is applied, and then she removes the stretch stimulus, and then the channels close. So these channels, when you put them in a membrane, they form functional, right, mechanosensitive ion channels. So this is all consistent with these channels then, right? They're within the sensory cells. They would sense, right, the stretching of the cell membranes of those sensory cells in the trigger hair. That would start that initial electrical signal, which would then be propagated by other ion channels through the leaf, right? But these would be the initial sensory proteins which cause that initial depolarization to trigger this electrical signal. So, right, they've got the right properties, they're in the right place. Are they actually involved in that touch response? To test this now, we turn to genetics. So we're trying to disturb the system. Usually you would disturb gene function with some kind of either a pharmacological approach, or in this case, we're going to go in and we're going to try and manipulate the genome of these plants to disturb the function of those genes. And the technique which we're using here, right? So this is gene editing now. We're using a particular technology which is called CRISPR-Cas9. Who's heard of CRISPR-Cas9? It gets a bit of press. Most people in the room, not everyone. It's a remarkable technology which has potential applications in medicine, certainly in the fields of genetics and academic science. It's had a huge impact on what we do. And the idea here, right? This is basically where we're going to introduce into the plant. So again, we're going to make some genetically modified plants. And we're going to have these plants make this protein, right, which is called Cas9. And basically Cas9 is going to come in and bind to DNA. So this is our DNA strand here. It's going to come in and bind DNA. And Cas9 is an enzyme which cuts DNA. And then what we also express in these plants is something to direct that Cas9 enzyme to the right location to cut at a very specific, precise spot. And these we call guide RNAs. So that's depicted here, kind of this blue-green structure here. And so part of this guide RNA you can see here is associating with Cas9. The guide RNA also has a region which we call complementary. Basically, it's specific to a particular part of the genome. So in our case here, we have a guide RNA which targets this DNA cutter to the flycatcher 1 and additional guide RNAs which also target to the flycatcher 2 gene. And once Cas9 is targeted there, it will cut the DNA. The cell doesn't like to have its DNA suddenly being damaged and cut. So these repair pathways kick in to try and correct that break there. And sometimes when they repair the DNA, a mistake occurs, right? And you generate a mutation within that particular gene of interest. So one of the nice things with, carnivorous plants, there's not a lot of labs in the world which are genetically modifying them, and there's a few reasons for that. One is they're very slow to grow, and there's a lot of challenges with these techniques. One nice aspect for them is that, and maybe many of you have done it, is that it's very easy to grow these plants in tissue culture. So what we can do is we can basically genetically modify just one cell, right? And then we can regenerate a whole new plant from that one cell, and then we can clonally propagate it out and then study those clonally propagated plants. So this is something we can take advantage of in the lab. It's a slow process, but it works. So here are these mutant plants, right? So again, we're using this CRISPR-Cas9 technology and clonal cultivation. This is the wild type plant on the left, and then in this case, it's a double mutant plant, so it has mutations in both the flycatcher 1 and the flycatcher 2 genes. So these code for two different of these mass gas-like channels. And then on the right, we're not going to go into the details of it, but basically I've drawn up two gene models here. We have guide RNAs targeting both genes. This is the DNA sequence here. If you were to go to look closely through these peaks, when we sequence the DNA of these mutants, you'll see that that DNA sequence looks different from the normal, what we call the wild type plant. So we've introduced mutations in these genes, which will disrupt the ability of those genes to produce the flycatcher 1 and flycatcher 2 proteins. And then the question is, right, are these plants any different? Have we disturbed their ability to respond to the touch of prey? And initially, I got to say, I was a little bit disappointed. So the mutants look normal. They look normal. On the left here, you're looking at in terms of the structure of the trigger hair. That wasn't surprising. I didn't expect them to. I didn't expect the trigger hairs to look abnormal in these plants. So trigger hairs look normal. The sensory cells are still there, have the normal shape to them. What you're looking at here on the right, though, this now is the percentage of leaves which are closing in response to what I'm calling now a harsh touch stimulus. So this is basically me just going in with my hand or a micro manipulator and giving those trigger hairs a little poke, like what I did here, right? And I give them 2 pokes within a 10-second interval, and then see if the traps close or not. And they look perfectly fine, right? So the traps, at least on a broad sense, seem to be functioning okay. What we did at this point, again, we're a little bit disappointed when we saw this, and a little bit surprised. We sat back and we thought, well, maybe what's going on in these plants. If you remember the earlier graph I showed, right, we identified at least 3 ion channels in these plants, flycatcher 1, flycatcher 2, which have mutated. There was also that Oscar channel, though, which is still functioning normally in the background of these plants. And there could be other ion channels which we just didn't pick up with our particular methods. So there could be, right, we knocked out these 2 and mutated these 2, but there could be other ion channels there in the background still within the sensory cells which are functioning okay and can still sense that harsh touch stimulus. So this is something in biology which we call redundancy in function, and it's particularly common in plants, which tend to have very large genomes. A lot of gene duplication occurs, and you can have lots of genes which are contributing to one particular function in this case. So we thought then, well, maybe what we need to look for is a more subtle defect, right? We've knocked out 2 amongst many. Maybe they can still sense a large touch stimulus. Let's go and do a very gentle touch stimulus and see if they can now sense that. And the method here which we turn to, we set up kind of a novel assay in this case. So we wanted something very quantitative. What we're doing now is ultrasound stimulation. So we cut these leaves off. We put them on a little bit of ultrasound jelly, all right, the same thing they use at the doctors, and we sit them on this little ultrasound transducer, which you can see here on the left. So this was a collaboration now with a group at Salk, which studies what they call sonogenetics, the group of Srikanth Chalasani. And then we can basically apply a mechanical ultrasound wave, right? So these are high frequency sound waves. We can apply this sound wave to these leaves, and we can gradually increase the intensity of that sound wave. And then we record the moment at which that sound wave, right, causes the leaf to close. So you can see this here on the left. I'm going to play the video, right? We're applying an ultrasound stimulus, and then eventually you reach this point which triggers the leaf to close. So we don't know exactly, right, how this relates to touch necessarily. It could be, right, just making that trigger hair rock back and forth. The ultrasound stimulus could be acting directly on the membranes. We're not sure entirely, but it is, again, it's this mechanical sound wave which is inducing the closure of the trap. And what we find now with this quantitative assay is we do see a defect. It's subtle, but there is a defect which we consistently measure within these plants. So it looks like, right, that these channels, as I said earlier, it looks like they're probably functioning in coordination with other ion channels on those sensory membranes to sense the touch of prey. So this is the model which we propose. So putting all this information together, right, here's our model for Venus flytrap touch sensation. We have our flycatcher 1 and flycatcher 2 ion channels embedded within that sensory membrane of these, right, of these sensory cells at the indentation zone. We also have the Oscar channel, right? We know that these channels aren't the only ones acting there based on what the plants look like when we mutate these channels. So there must be other sensory channels there too. There could be others we haven't identified. And then what happens when an insect bumps this, right, the lever of the trigger hair, it bends those sensory cells, you get membrane stretch, it opens these channels, and then you get the movement of various ions, at least for the Fly C1 and Fly C2 channels. They're mostly conducting chloride ions. So these are, in this case, in plants, they're moving out of the cell. This causes that change in charge across the membrane, and then other ion channels kick in, right, to propagate that change in charge through the rest of the cells of the leaf, right? And that's what we were measuring earlier. And then you get leaf closure. And finally, again, to just reiterate this point, what I think is kind of interesting about this study is it does strengthen this notion in evolution, right? I mean, how do you get these crazy, bizarre plants, right, which do these remarkable things? You know, it strengthens this idea in evolution of co-option. In this case, we're taking genes, proteins which are already out there. In bacteria, they're sensing osmolarity, changes in salt concentration, and basically we're taking these genes and putting them to this new purpose. In this case, right, coupled with this architecture of the trigger hair, we get this exquisite touch sensitivity. So this is how you can evolve this this touch sensing structure, right? Take these genes and put them to this purpose.
Venus flytrap cultivars that like don't shut?
Yeah, there is one which is studied, you know, you read about them. I don't have access to any of them. There is a group in Germany which has studied one. It doesn't, the defect though is not at the sensory level. It's somewhere downstream of that for why it doesn't shut. So you can imagine that whole process, right? You've got the process of sensation, which is what we're looking at here. And then beyond sensation, right, you've got to propagate that electrical signal. So there could be a defect which occurs there to prevent the leaf from shutting. And then it's also got to trigger some kind of changes. It's probably changes in turgor at the midrib to then cause that bistable, right, buckling movement. And you could have a defect there as well. So to get a trap which doesn't close, you could picture that there could be a defect at multiple points. But yeah, I mean, you know, obviously this is a hobbyist audience. One of the great things about carnivorous plants is that there's a lot of hobbyists who love to grow them, and you guys have generated some pretty weird, right, carnivorous plants out there. You guys are laughing, but it's true. And you brought up one. Unfortunately I don't have access to all of these. There's a lot from Europe, but that is one approach which scientists are taking, is to try and get access to some of these weird Venus flytraps. There's one out there which doesn't have trigger hairs, for example, right? So it's a developmental defect. And to basically take these mutants, which hobbyists have generated, and then to use them to try and work out at the molecular level what's going on, right? To take the mutant and try to identify the gene which causes that particular appearance of the mutant. So that's another approach which other groups are taking. It's a good question. And then finally, I return right to this big picture question of mechanoperception in plants, right? What we've learned now in these carnivorous plants, does it apply to some of these other processes of mechanoperception, which we see in non-carnivorous plants? I already brought up that these mass gas, right, these flycatcher-like channels, they're probably regulating cell homeostasis. They're involved with pollen hydration and protecting the pollen cell there. You brought up, someone mentioned the piezo channels. So there are other mechanosensitive ion channels. The piezos seem to be involved with root movement. You know, what are the ion channels involved in these processes? Is what we're learning in carnivorous plants, is some of this conserved across these other plants? So this is a question mark, which other groups are still exploring. And then finally, this is my last slide before the acknowledgements. I think it's an interesting slide to end with. I mentioned that ultrasound assay, which I did, was in collaboration with another group at Salk, the group of Srikanth Chalasani. So what his group is interested in is taking these mechanosensitive ion channels and actually putting them in human cells. Right, so you put them in an animal cell, and then you can apply, right, they're sensitive to ultrasound. You can apply ultrasound stimulation to those cells, and basically you can make these channels then open and close in a controlled manner, and you can change the properties of those cells and their activity. So you can imagine in something like medicine, there can be, right, you can have an insulin secreting pancreatic cell, you can have neurons, right, which are behaving aberrantly in some part of the body, and you can start to put these channels in and then regulate the activity of those cells with ultrasound. So it sounds a little bit sci-fi, but it's cool to think about. So we actually have a patent on this technology. These plant channels have some interesting properties to them, which are a little bit different from animal mechanosensitive channels, right? You brought up piezo. They're different from piezo due to the ions, the salts which they conduct. So they give us a useful tool for these kind of techniques. So again, it's a slide I like to end with because I think it's kind of cool to see what we learn from plants. That, you know, you never know how these things might be applied. Thank you.
Have you tried optimizing the frequency for response?
So those are experiments which are being done by Srikanth's group. So there they do look at, you know, they need something. If it's going to go into tissues, they need, right, particular ultrasound properties which can activate and close these channels. So this is something which they're playing around with and looking at. Yep.
Quick question.
Yeah.
curious. The cells that the trigger cells, for example.
Yep.
Those cells, are they any way related to, for example, a sunflower? Sunflower follows the sun during the day.
Yeah,
Is there any relation to that?
I mean, that's sensing a different environmental input. So there you're talking about light sensation. And then as far as I know, in the example which you brought up, although again, I'm not an expert in it, but as far as I know, that's a growth response, right? So then you're getting differential growth in different parts of the stem. So think about phototropic bending, for example, right? Where a stem bends towards light. This is a differential growth on one side of the stem versus the other. So that's different than what's occurring here. It's certainly still a response to the environment. In this case, light. Light is not. You don't get these electrical signals occurring. So there's different proteins involved. No. Yeah. No. So different, but and again, you get this developmental response here with the Venus flytrap. At least, it's a bistable, right? It's the leaves, once they start that movement, it's bistable. The classic example, which I always think of, is if you get like two sheets of metal or card or something, right? One's a little bit bigger than the other, and you glued them together, you'll find that they will buckle, right? Once you start moving them, they'll snap in the other direction. So that's kind of an analogy you can draw with the Venus flytrap leaf. Mimosa, yeah. So that is electrical signaling. I don't know enough about how the movement occurs there. As far as I know, there are motor cells in that example. So I think that's a little bit different. But again, don't quote me on that. But it certainly uses electrical signaling. Yep. Very quickly to thank the people who have helped with this. Firstly, a shout out to my boss, Joanne Corey. She's allowed me to do some experiments which are definitely out of the mainstream of my lab. My lab mostly does study light responses. So she's been very generous to help support this particular research program. These are my other collaborators I would like to acknowledge, in particular, Ivan and Liz Haswell, previously from Washington University. So they helped with the calcium imaging experiments, which I showed of Sunju. A huge shout out to Ardem at Scripps here in San Diego, and in particular, one of his postdocs, Swetha Murthy, at the time. So they were really instrumental in driving this Venus flytrap work. Srikanth and Wenmei, in Srikant's group, who helped with the ultrasound experiments, and then Sergey in my lab, and Charlotte, who was a high school intern who worked with me, who helped develop the methods to generate genetically modified carnivorous plants. This is my funding, NIH. I have a Salk Innovation Grant. Private donation from Howard Newman has funded other aspects of my work, and then also the Howard Hughes Medical Institute. And yeah, I'm happy to take more questions. Thank you.
So I keep reading about climate change and how carnivorous plants are sensitive to the changing environment. I know you said that your species is mostly light and how it affects plants. Do you know of any other species that might be using like CRISPR to make these more adaptable or durable?
Yeah, so this now is going obviously beyond carnivorous plants. This whole concept of resilience at the moment is of huge interest to the plant biology community. Obviously, in terms of studying natural ecosystems and how they might change as climates change in the future, but also a more applied aspect to that is, can you make more resilient crops, for example? So yes, these technologies, right, CRISPR-Cas9 technology, this is being used for these particular applications as well. It actually, there's some interesting aspects to CRISPR-Cas9 from a regulatory point of view, in that you can put these artificial genes in, you can cause mutations in the genome, and then through standard breeding and crosses or other methods, you can then essentially remove those artificial genes and just leave the genetic lesions behind. So from regulatory point of view, right, how you regulate that when there's no artificial genes in there, it tends to be much easier to get it to market. So there's lots of advantages to CRISPR-Cas9. It's a wonderful system. Yeah.
Yes. I actually work with the Crops Initiative at Salk.
Okay.
So we are working with crop plants to develop like more resilient crop plants, whether that be like drought tolerance, salt tolerance, but also aiming for like deeper, bigger roots that take down more carbon from the atmosphere, since most of our land mass is used for crops. So Salk is definitely doing a lot with.
Yeah, there's a lot of interest in this at the moment. Yeah. Thank you. Yeah.
CP question. You worked with sundews, Doceria, anyone do anything with bladderworts? Really quick responses.
With bladderworts, yeah. So there's I don't work with bladderworts, and they're obviously, I mean, they're faster than a Venus flytrap. There is a group in the UK which does very nice work with bladderworts, and they are mutating genes in bladderworts as well. So the answer is no, I don't work with them. They are a unique system. They're a little bit of a different system though, in that they're right in these plants in the Droseraceae, you have these electrical signals which are inducing the movements, whether it be that bistable buckling or some kind of, it's not quite clear what's going on in Drosera, but let's say it's a growth response, right, of the leaf growing over the prey. In the bladderwort, to get that really rapid movement, it's all mechanical. So I like to liken it more to a mousetrap, right? A mousetrap is a mechanical structure. There's no electricity or anything required for it. Basically, there's a lever and a catch, and you trip it, and it snaps closed. So that's kind of an analogy for a bladderwort. You don't need those electrical signals. At least no one's seen them for closure to occur. So a little bit different process.
I know genetically, let's say they have a smaller gene.
Yeah, there's this argument for some carnivorous plants. I can't comment on how true it is, but certainly some carnivorous plants have a very small genome, and the argument for that is if you're living in these nutrient-poor environments, nutrients are limited to you. Your genome is DNA, which is made from things such as phosphorus, for example, right? You got to put atoms in there. So therefore, you have a argument is that you might have evolved a very small, compact genome, so that way you're putting less nutrients towards making the genome. So that's the argument with these kind of plants. But again, not all carnivorous plants fit that idea. So, any other questions? Yep.
I had a question. I was curious about the process from when you guys kind of figured out that you wanted to look into the Fly1C and the Fly2C ion channels, and then when you went into the time it took to then go in and mutate the genome and then to grow it in tissue culture. What is the kind of time frame?
You're talking years. So the question was how long does all this take? And it's years. Or I'm guessing in this room, probably everyone has grown a Venus flytrap, and they're not the fastest growing plant. So that takes time to firstly just the initial science experiments, which are the discovery phase to work out what's there and what's interesting. But then the longest step is to, right, as I said, you try and genetically modify essentially just one cell or a pool of cells, and then you've got to grow out whole new plants from that. And that's a lengthy process. So it takes years. And that's, I think, one of the reasons why there's very few labs in the world. I mean, part of it's obviously funding, and these are unusual, interesting plants, but they don't, because tends to be not an application, right? Although I tried to show you some potential applications today. So one is just having the community around them to push along the technology, but the other is there's limitations just due to the inherent nature of the plants and how long it takes to grow them and work with them. Thank you.
Another round of applause for our executive.
Thank you for that wonderful presentation and a few housekeeping things before we wrap up, before I let you guys go and we'll get on with the rest of our program is the next couple of meetings. So the next meeting right after this is the LACPS meeting. If you're ever in that area, they're having a general regular meeting on February 17th, a Saturday. And then our next meeting, the SDCPS regular meeting, is going to be Saturday, March 23rd. So mark your calendars. It'll be in this room again. And we don't have a program scheduled yet. But if you have any interesting presentation ideas, it doesn't have to be something like this. It can be something more casual. We are open to any people who are willing to volunteer and present on any kind of interesting topic. Or if you have any ideas of what you want to see or hear about, please let us know. And then we might find someone who fits that niche. See. That should be it. Yeah, just be on the lookout for our next regular meeting. And if you're also up in the Orange County area, there's also the Skippy Club, the San Diego Carnivorous Plant Enthusiasts. And they're also having a meeting sometime in March, details to be confirmed. Be on the lookout on the Facebook group or the email list. I'll be sure to send that out. Thank you. Yeah, you had a comment?
I guess with this meeting type things that everyone who should get a free raffle ticket. We'll be doing the raffle soon. So if you receive the raffle ticket yet, let me know.
Yes, please.
Discounts at your
Yes, as a reminder, we will be having the raffle soon, so get on that.
And I'd like to throw out one more comment for the kids in the room. I have some extra butterworts at the front, so if you want to come grab yourself a butterwort and try to grow it, you're welcome to do so.
Fascinating talk.
Thank you
very much.
Thank you.
I love your shirt, by the way.
Yeah. You want a butterwort? You want to grab yourself a butterwort?